I'll be talking about Mendelian speciation, but it will take us into the area of intelligent design and irreducible complexity and all that kind of stuff. So it's rather fascinating. Let's see now. This paper came out. It came out about a month ago, and I was very surprised by the first comments on this paper. It's from one of the most prominent science journals. It says phenotypic evolution, and if you prefer you can just read evolution there, was long perceived as a slow process driven by gradual accumulation of new mutations. And basically what that's saying is everyone thought, you know, the world can sort of mutate into existence or whatever, or at least the animal life can, etc., etc. And then they went on to write, however, recent studies across diverse organisms show that the reshuffling of already existing genetic variation, there's someone shuffling away there, whether standing within species or introgress from other species, pre-existing variation provides an important fast track to phenotypic evolution. It might sound like a complicated statement, but basically what they're saying is Mendelian speciation, what we spoke about before, is the way this is going on. The information is already there, it gets shuffled about, and because it gets shuffled about, you get new species. And Mendelian speciation is obviously named after this guy, Mendel. He was an abbot at the end of the day, but he was a friar, an Augustinian friar. And he studied peas. And he looked at various features of peas. He looked at flowers, and he looked at seed colour, and he looked at whether the seeds were round and smooth or wrinkled, and he looked at the pod colours, whether they're yellow and green, or the pod shape, whether inflated or constricted. He also looked at the position of the flowers and the plant height. Now when I use the term phenotype, I've used it a lot, all that basically means is what you're looking at. So if it's flower colour, it would mean purple or white. Or if it's seed colour, it would be yellow or green. And we all get two chromosomes. We get one of these from mum and one of them from dad. And therefore we have possible traits. So Mendel, when he was working, he crossed purple flowered peas with white flowered peas. And he just got purple peas. It was as though the white had vanished. There were no white flowers anymore. That was very sad. When he took those purple flowers and crossed them, then suddenly he got white flowers again. They could be hidden in the genome. They could be latent. They could be in the chromosomes. The information could be there, just not expressed, not shown. Now that has all sorts of consequences. First of all, just briefly, he had, because purple always showed up, that was dominant, and the one that didn't show up, that was the white one, that was recessive. Now, under those circumstances, he was looking at just one character, it was flower colour, but he could see two things. They could be purple or white. When he extended that to look at two different things, like seed colour, which would be here yellow or green, or seed shape, which would be wrinkled or smooth, you can see these are smooth, and these two here are wrinkled. Now we can get four. This is a picture of some studies. He didn't just look at peas, he also looked at beans. And if he looked at three characters, he could get 1, 2, 3, 4, 5, 6, 7, 8 different phenotypes. So this is grand, yeah. And quite obviously, as the number of genes increases, even if it increases linearly, the number of phenotypes you get, that increases exponentially. Mendel looked at 7 characters. I showed you a picture of those before. And that would mean have been able to see 128 different phenotypes. And he reported seeing them all. Yeah. It's very obvious from this that this is where biological diversity comes from. When we look around and see all the diversity in the world, we don't need any mutation events. All we need is 2 copies of each gene, and we get one from mum and dad, one from dad, so we already have that. If we look at 20 characters, suddenly we can generate, because it goes up exponentially, so we can get 1 million different phenotypes. Yeah. So it's absolutely insane how quickly and how much diversity is present inside, you know, our genomes. Now, in my title, I spoke about characters and traits. So I do have to very briefly mention what these are. Yeah? It's really critical to teach students of genetics the difference between these two. Why is it important? Well, traits, these are the things you see with your eyes. Yeah. They can be made by mutations. But characters can't. And I hope by the end of this little presentation today, you'll feel very comfortable with that comment. Characters and traits, they must be distinguished. All too often, characters are just carelessly called traits, but if they are, then traits have to be called something else. They have to be called trait versions. Now, do not worry, I'll have plenty of examples to show you what I'm talking about here. Yeah. But it's vital that the two terms are kept separate. We should avoid ambiguity. They refer to different things. Characters are general attributes of phenotype. Things you see, I mean, we already mentioned them, like flower colour or pod colour. Now that is a character, but it's not actually a colour. It's talking about colour. That's the character. And then we actually have the traits, which will be purple or white or yellow or green or whatever. That's what you actually see. There's a difference between them. And what speciation does is it takes each one of these 7 characters, and we've got different options here. So we got purple and white. They're the actual traits. That's what you actually see. But there's this category, this concept of flower colour, and that is the character. And as you can see, although you've only actually got technically 14 alternative traits here, you can make with this very comfortably 128 different combinations. Mendel actually understood more about it than just that. He understood that these characters could result from, well, sub-characters. There was more to it than just one single thing. He wrote in his paper, his terribly famous paper, the colours of flowers and seeds of beans is a combination of 2 or more entirely independent colours, which individually act like any other constant character of a plant. So up to this time point, these things had been called, this had been calling A and B and C. But now he started to talk about A1 and A2 and A3 because there was more parts to it. He intentionally labelled these sub-characters this way. He wanted his readers to understand they all contributed to the same character. Each sub-character still had 2 traits. And this was just a hypothesis at the time, and he recognised it as such. He says, characters are features of phenotype. They arise through the action of multiple genes, are polygenic. It is probable that in this way we might learn the significance, the extraordinary variety of colouring of ornamental flowers. If you think about flowers and all the different colours they have, particularly ornamental flowers you buy, he was trying to answer the question, where do all these different colours come from? There's obviously more than 2. In fact, there's lots and lots. And at the time, he didn't understand how that came about, but he thought it would be via this mechanism. And in fact, it's gone on to be shown that's the case. So these characters then, they're not just a single thing, they're a complex of things. The characters arise through the activity of multiple genes. Obviously they're protein products, genes make proteins. And then their interactions, their intricate interactions. Well, that's one guy, and his name's Mendel. He's another famous character out of history, William Paley, and he brought the watchmaker argument, and I'm sure many of you will have heard of the watchmaker argument. It's all based on teleology. There's the idea that there's a purpose behind things which you can recognize if it's been made by an intelligence. So if you find a watch, you don't just think, oh yeah, that's a rock. You think, oh, this is actually a mechanism behind this. This has been designed. And one of the most important features, there are aspects of that design, is something called irreducible complexity. So I'm going to show you another picture of it's sort of a watch, not quite the same thing as a watch, but this comes from 100 BC. This is the Antikythera astrolabe of Archimedes. What on earth is an astrolabe? Well, it's got astro in it, it's got something to do with planets and things. Actually, this thing here, it could tell you where all of the different planets are every night of every month of the year and all this kind of stuff. Yeah, very fascinating, from 100 BC. And when they originally found it, it would just look like junk. Yeah. When they studied it, they realized it's not junk, it's not just rock debris, there's a purpose behind this. In fact, it's got all these little cogs and stuff in. Yeah. It is what is called, or referred to as, irreducibly complex. What does that mean, irreducibly complex? So I got this little acronym here to help us with it. It's MIP, M-I-I-P, and that simply means many interacting, indispensable parts. You have to have them all. You can't lose one or the other, because then it won't work, and it will be useless. So if you have them all, and you've got irreducible complexity. Now Mendel's choice of subjects, these various characters, they were wide-ranging. There was a wide variety of them. And his study included, say, these 7 characters, but it was basically a cross-section of plants. It looked at seeds and flowers and fruits and stems. It looked at the response of the plants to light, or the response of the plants to plant hormones. Yeah? It looked at tissue determination and organ placement, all of this. Yeah? And if any observations were true for these characters, for all of these characters, then it would basically be an essential truth for all plants. And by extension, presumably, there would be a lot of truth for that for all animals too. Speciation exploits the fact that characters have dominant and recessive traits. Dominant, remember, they always appear. Recessive can be hidden. And species are unique, fixed combinations of these. So a species might have purple flowers and green seeds and wrinkled seeds and green pods, but another species might have white flowers, perhaps green seeds, but then round pods, and they're just different combinations. Yeah? And a question arises, how do dominant and recessive traits differ genetically? And does it say anything about the source of the information? Is there evidence, perhaps, of 2 sources? An original source, primary traits, and then a derived source, secondary traits, which came later. Characters, these are technically the building blocks of the organisms we see. A fancy way of saying that the building blocks of phenotype. Yeah? They have to be generated, and the other thing that happens, they have to be regulated. You can't have arms and legs appearing in the wrong place at the wrong time. That would never do. We need to have sensible organisms. Yeah? Dominant traits reflect a synergistic, purposeful expression of these characters, genes, and proteins. Logically, dominant traits result from the successful expression of pre-existing genetic programs, and that's just a complicated way of saying the information which is in our chromosomes is there to make something. And the dominant trait reflects that. It's the reason why it's there in the first place. Recessive traits, they result from just tweaking it a bit, altering single components. We don't need all of the components, we've already got them with the characters. We just need to tweak it back, a single one. Sequence analysis of Mendel's 7 characters. We'll see that they repeatedly demonstrate that recessive traits arise from just single changes within something which is irreducibly complex. Well, this year, in May, a remarkable paper appeared in another famous science journal. You can ask my geneticists, genetic students about famous science journal. It's one called Nature. And a remarkable paper by a guy called Feng. 47 authors were named, and that's becoming more and more common now with these huge studies they do in genomics. 47 authors. There were doubtless lots of other people being named, probably twice as many, and so we're trying to fight to get onto the paper. And they no doubt contribute. The research lasted 6 years. And they sequenced the DNA of 700 P genomes. Now, when you sequence DNA, you have to make sure you're right, so you repeat it. So for every one of these 700 P genomes, they actually sequence 20 times. They get 24 coverage. Extraordinary research that had been done. And the purpose was to determine the genetic differences, these alternate traits of these 7 characters of Mendel. So there he is, 1866, finished his paper. So what did they see? What kind of changes can take place to our DNA? Well, the 3 main ones they found. There's one called substitutions, and most of you, I think, will be familiar with these. So you've got this sequence, A's and T's and G's and C's, and you can tweak one of them. So instead of having a C, you can have a G, or instead of having a T, you can have an A. Well, that's a substitution. But there are other things you can have as well. You can lose a chunk, and that's called a deletion. Or you can stick a chunk in, and that's called an insertion. These insertions are quite intriguing. They're sometimes called transposons and things like that. Well, that's what we got. Those are the kinds of changes that you can see. Now, of course, just to say that doesn't really cover what's going on here, because genes are complex. And depending on where you tweak it and where you poke something in, you see this red bit of this gene, that's the bit that's going to make the protein at the end of the day. And all the rest of the information at the front and at the back, that's to regulate it so it switched on at the right time in the right place. And there's also some funny bits here, and people don't really know why they're there. They're called introns, and they get cut out. And I'm sure someone's going to get a Nobel Prize for working out why that happens. We know a little bit about it now, but not very much. Very, very fascinating. And these substitutions, deletions, and insertions, they're happening in this. We already have something incredibly complex. If you start to tweak that, things are going to happen. Mendel selected his 7 characters, and he gave them names. And he based those names on his observations. So the terms he used were practical. This. But actually, it missed out on the fundamental biological processes involved in the production of the characters. So let's check out these 7 characters and see what we learn. The first one was seed shape. You can see this one's round and this one's got wrinkles in. In fact, the wrinkled one turned out to be quite popular because it had a higher sugar concentration, so they were sweeter. So people like these wrinkled peas. But what's actually going on is something called starch storage in the seed leaves. So what are the seed leaves? Let's move over. If you take a pea and you crack it open, there's 2 sort of thick leaves in there. They're called seed leaves. And they're there to feed the little baby plant, because it's a seed. And the way it's fed is there's starch in there. And what's interesting is this starch molecule. It's a fancy molecule here, a starch molecule. And you see that here it's sort of straight line, but here it's branched. And the starch should be branched. If it's not, if it's straight like this, then instead of getting round peas, you get wrinkled peas. So that's the difference, yeah? Many proteins are involved in this process. The synthesis of starch, of branched starch, requires starch synthetases, ADP, glucose, pyrophosphate, starch branching enzyme, starch debranching enzyme, kinases, all sorts. Woo! Yeah. Here's a more detailed picture of what's actually going on there. And each one of these little green asterisks is there. That's protein. That's made by a gene. This is how we make starch. Starch biosynthesis involves the production and regulation of loads and loads of proteins. It's an irreducibly complex process. Oh, if I go back to it, I highlighted this one. This is the one that's going to get mutated. Now, as long as it works, you get your round peas and everything's fine. But if you mutate any one of these, then it's not going to work anymore. And that's exactly what happens here. Here's the gene, and this is the normal one. It's on chromosome number 3. And here's the gene with the mutate. There's an insertion in it. It doesn't work anymore. Non-functional protein. And what happens? Well, the seeds have abnormal starch and twice as much sucrose in, which is great, but all that sugar draws water in. And so the peas get big, and then the water goes out again, and it shrinks, and it leaves them wrinkled. And that's why you get this wrinkled stuff. We have wise design here. The ability to store starch reserves in pea leaves, that's obviously important for the growth of the pea embryo. But to get all of that stuff, what would you need? You need an unfathomable number of beneficial mutations. There's no way that process is going to mutate into existence. Yeah? Alternatively, it provides compelling evidence of design. And one thing we do know for sure is mutation actually compromises the whole. Mutations are an unconvincing cause to account for the existence of the intricate molecular biology of starch storage in seed leaves. Mendel also looked at seed colour. You get yellow seeds and you get green seeds, and what's going on there? It's got a lot to do with chlorophyll degradation. Chlorophyll degradation in seed leaves. Chlorophyll, of course, that's how plants capture light. Chlorophyll pigment making plants green. It's a compound molecule with a porphyrin ring and it's got a magnesium ion in there, and it's got a little tail here. I'll tell you about that later. Hydrocarbon tail, called a phytol chain, whatever. And together with other pigments, it can capture light. But this is ultra precise in the way it works. Yeah, definitely evidence for design that goes beyond all reasonable doubt. This is the light harvesting complex. It's made of 3 proteins with their pigments attached. They've been labelled here 1, 2, and 3, and if you just take one of these out and look at it, because they're identical, you can see the different pigments. They're sort of the colours of light which they absorb is sort of shown by the pigments. There's chlorophyll here, and then there's some other pigments there. These all have to be at exactly the right angle. In fact, the whole unit won't work unless 3 of them line up together and click in. And then as soon as it does, boom, the thing captures light. We do not have the technology to make anything like that. Anything. It's just absolutely extraordinary. Here's that chlorophyll molecule down there. There's a tail, and there's the bit that captures the light. There's a magnesium ion in there. If it doesn't have that magnesium ion, it's called pheophytin. And these babies, they're used to catch light. Light comes in, and then it bounces around these pigments. They have to be all exactly right. It's not going to work, yeah? They bounce around these pigments to this reaction center. And then the energy is added to an electron. The electron comes high-powered electron. It can do its stuff, and it's great. Here's another picture of that region, the reaction center. And this captures the light, and then it adds that light energy to the electron, and it gets bounced from these chlorophylls to these that don't have magnesium into this stuff, and then it's passed on. But the electrons have to they have to come from somewhere. They come from water. And an electron stolen from water, and then another one is, and then another one, and then water falls apart into hydrogen and oxygen. And hydrogen is used to make sugar, and the oxygen the plants breathe out. And that's why we like to go for walks in woods, because there's plenty of oxygen around. And it's because of this process here. There's a lot to that process. Chlorophylls' light capturing properties cause it to be phototoxic, and therefore many fruits, when they ripen, they degrade the chlorophyll, because you don't want something which is toxic in you. And in fact, because the chlorophyll is degraded, the fruit changes colour, and instead of being a green colour, it goes to a yellow colour. I don't know if you have any sharp sighted people notice what happens to their green bananas if they wait long enough. They start turning yellow. Well, it's to get rid of some of that toxic chlorophyll. Loss of the green pigment, it acts as this visual signal then to fructivores. Very helpful. These animals that eat fruits, even folks like us, we know it's ripe when it's no longer green. The first step of this degradation is using this protein to make pheophytin. Doesn't have the magnesium in it. So enzymatic chlorophyll degradation requires at least 5 proteins, and then other proteins are required to make sure that it happens in the seeds, and only when the seeds are mature we have irreducible complexity here. Now, yes?
I have a question about the irreducible complexity idea. These systems obviously differ in the number of components they have in them, but is it generally true that if you remove one component from that system, it will no longer function?
Exactly.
Okay.
Yeah. The parts are indispensable. You need them all at once. Yeah. And what happened then in Mendel's trait here? Why has it stayed green? Why is it not going yellow? Well, it also got, in one case it got an insertion. In another case, there was a deletion. But in either case, this DNA complicated itself because of this change, this mutation. It doesn't work anymore. Yeah? And if it doesn't work anymore, then this is going to stay green. Chlorophyll degradation in seed leaves involves a large number of interacting genes and proteins. It's irreducibly complex. The ability to effectively and selectively degrade chlorophyll in pea seed leaves would require an unfathomable number of mutation events. And they'd all have to be beneficial, because of course if they're not beneficial, that's no good. Alternatively, it's clearly logical evidence of design. What about flower colour? We like different coloured flowers. Well, this is fine. So what about this one? Actually, what's going on here is something called anthocyanin biosynthesis. Anthocyanin, fancy name for a pigment. And it's made in flower petals. There you go. There's some flower petals. You see, these are purple and these are white. These ones that are purple, there's the one that the pea flowers, lovely flowers. Lovely flowers. They have a big petal at the back called the standard. And then 2 on either side called the wings. And 2 which are hidden, actually fused, and then they hold the stamen and stuff inside. Very fascinating flower. Mendel, when he was doing his studies, he bought the seeds. And there's a difference in the seed colours here. You can see this one's different from this. This one gives purple flowers and these give white flowers. So he spoke about seed coat colour. But everyone nowadays talks about flower colour because people like flowers and think of flowers more. But it's the same that's the thing which is going on here. And the problem is that getting that purple pigment. Pea flowers should be purple. And if that pigment isn't present, they end up being white. But white isn't a pigment in this case. White is just something which happens. It actually results from the absence of the pigment. Purple plants synthesize this pigment to be purple, anthocyan. In most white plants, there's no white pigment. Instead, simple light backscatter from the petals. It's perceived as white. If the pigment of that was there, it would be filtered by the pigment. It would go purple. So anthocyan pigment biosynthesis. How's that controlled? Well, there are 3 proteins that do that. They're very special proteins. They switch genes on, lots and lots of genes. And some of these genes here tell the pigment to be made in those back petals and some in the side petals. But we're interested in this one because this is the one that gets mutated. If you want to make the pigments, it's not easy. You need lots and lots of buddies to help. You see these little green asterisks? They show you all of the different proteins associated. And there's lots of them in peas. Numerous genes switch on to make numerous proteins, and they will turn phenylalanine, that's an amino acid, they'll turn it into the pigment. But this is an irreducibly complex process. Now here's our normal gene, and a substitution took place, a G to A, and it took place at a very critical point called a splice site, whatever. But it meant that this protein isn't made anymore, and if the protein isn't made, the pigment isn't made. That's why you get white flowers and not purple flowers. In order to make and locate the anthocyanins, the whole process would require an unfathomable number of beneficial proteins, loads and loads of them, because you need all the genes and the proteins, and you have to enable all their complex interactions. Of course, the alternative is that it's compelling evidence of design. Yes?
Another question. In regards to beneficial mutations, I researched at one time trying to determine how often beneficial mutations take place, and the sources I found estimated that it was 1 in 1,000 that would be considered a beneficial mutation, the other 999 of that 1,000 being either neutral or harmful. Is that, do you know offhand?
It's a very complex question.
Okay.
Do you like white flowers?
Yes.
Then this is a beneficial mutation. That's why it becomes tricky. So and we'll come back to this, and you'll see as we go through the lecture that you actually get sometimes nice things. We had sweet peas. People like eating sweet peas. It's not the best way for things to happen in the wild, but obviously if a farmer's looking after the peas, then he's going to grow them like crazy. Very interesting question to ask. Mendel looked at pod form. So he has inflated and he has constricted. What's going on here? There's a cell type, a tissue within the actual pod, which is responsible for this. By the way, the wild type, they're always inflated. We should have that. That's the dominant feature. That's what should be going on. But if we want to not just eat the seeds, but we want to eat the pods as well, then we want this mutation to occur. So is that beneficial? It's a very interesting question to try
marketable.
It's definitely, the Chinese are actually very gracious. They gave us our sugar peas. What they had to do is, inside the pod there's this layer. It's called the parchment layer. It's a tissue inside the pod, and it causes the pod to burst open so the seeds scatter. Well, if you don't have it, then the pod doesn't burst open, but The farmer likes that because he can sell these pods because they taste good. So the farmer keeps them alive. In the wild, they wouldn't survive very well. And this is difficult because this is looking at the cells now, but this is actually a cross-section through the pod, and you can see this big fat layer. That's the parchment layer. And if you look in these other situations, it's nowhere near as big, and that's because of mutation. And if that happens, then you get constricted pods. They won't burst open, so the farmer's going to have to come along and sow those seeds. But because they're soft now, they don't have this hard layer inside them. They're edible, and so they taste good. For a long time, it's been known that there, in this case, are two genes which are responsible for this. They're called one's called the P gene, one's called the V gene. And the formal name of the P gene is this complicated thing, and the formal name for the V gene is this complicated thing, and they're on different chromosomes. And one encodes a signal that tells the parchment where it should grow and where it should appear. You obviously need that. You can't have parchment in the wrong place, so that would just be nonsense. And then this one actually switches on the genes to make the proteins to make the parchment layer. And how are they mutated? Well, this P gene, that's just a substitution, so an A turns into a T, and it doesn't work anymore. And here, this is the normal gene, and suddenly this thing's got stuck in it, this insertion, it got stuck in, and now that doesn't work. Now they don't work, and now you get constricted, or technically down here, constricted pods. Mutation events are an adequate explanation for the origin of this tissue. Many genes and proteins are involved, and lots of intricate interactions. This is irreducibly complex. Such elaborate and complex cellular processes, they supply very compelling evidence for design. But you're going to have a dickens of a job of mutating that into existence. We got pod colour, and pod colour reflects P-pod chlorophyll biosynthesis. Now, the last time we looked at chlorophyll, it was degrading it. Now we want to make it. This is what green chlorophyll looks like, the molecule. This is the bit that absorbs the light. There's the magnesium ion inside. And this is the tail. The tail is very important because it holds it in the right place at the right time. How difficult do you think it would be to make chlorophyll, a molecule like that? Its synthesis, its biosynthesis, very complex. It starts with 9 proteins, 9 enzymes, and they start from glutamate and then end up making this thing. And then 6 additional steps actually culminate in the chlorophyllase. You've got 15 different proteins here. Chlorophyll A itself is phototoxic. So even more enzymatic steps, there are more proteins are needed to get it into the light harvesting complex so it's not dangerous. All of that. Woo! Here's a little picture. And here you can see these little red crosses. These are all the proteins you have to make at the right time and at the right place, otherwise you won't end up with chlorophyll. The one that we're particularly interested in here, I've highlighted it with these red arrows. This one's the one that's going to get mutated. And if it gets mutated, how will it get mutated? Well, when they look to see what had happened, this is the normal gene. Well, actually, this is the normal gene. And it's got some buddy genes here at the front, and all of them get cut out. And so this gene comes under the control of this gene, and far less of this gene is present, and that's why you get a yellow pod here, because the chlorophyll isn't made. This is a lot of complexity. The process of chlorophyll biosynthesis requires precise expression and production of many genes and proteins, irreducibly complex, innumerable mutations. It's absolutely impossible for that just to arise on its own. It's persuasive evidence of design. The one character I have been waiting to tell you about is this one. Now, technically, when it doesn't work, it's called fasciation. but this one is fabulous. So this is our normal pea, but sometimes it's grown in this state where the flower sort of bundles at the top. And it's all to do with flower regulation. Now, flower regulation is caused by something in a plant called the shoot apical meristem. Sorry that that's not quite of a name for it, but it has all sorts of interesting repercussions. If you look at this dandelion, do you notice there's something wrong with it? It's double-headed. And if you look very carefully at the stem, it's kind of hard to see because it's two-dimensional photograph, but it's actually too thick. Yeah? Now this is very interesting. Here's a little daisy. Can you see this daisy flower? Does this daisy flower look right? No, it doesn't look right at all, does it? There's an unspoken expectation of elegance in nature. We demand things to be right. It's very fascinating. And if it's not, we'll comment on it. Oh, well, it's not right. There's something wrong there. Yeah. The fascinating balance in nature. Yeah. And it's certainly by flowers, the control and regulation of all of this is by this shoot apical meristem. Now, I don't want to keep saying shoot apical meristem. It will drive you guys nuts. So I'm going to call it by its abbreviation. It's called SAM. Shoot apical meristem. SAM. Yeah? And SAM regulation is the glory of plants. It really is. It defines the architecture and all the leaves and everything. You have the shoots. Very, very fascinating. Yeah? Everything above ground. SAMs, they carefully regulate the proliferation, differentiation of stem cells. They define where a leaf's going to appear, where a branch is going to appear, where a flower, even a whole flower bunch of inflorescence will develop. And so, of course, SAMs are responsible for all of this. God so arrays the grass of the field. When we see all the beauty of flowers, it's natural to be amazed, even awed at the wonder of it all. Flowering plants are called angiosperms, the fancy name for clothed seeds. I'm sure the guy that used that term was a Christian, he was a German botanist. And it's all to do with the shoot apical meristem. It's to do with the fact that a flower is made, and the flower hides the seeds, it protects it. That's not the same in pine trees. It's not the same in other kinds of plants. But what is very interesting is what our Lord says about it. Our Lord Jesus spoke of the clothing of plants. He said, observe how the lilies of the field grow. The lilies of the field. Observe how the lilies of the field grow. They do not toil, nor do they spin. Yet I say to you that even Solomon, in all his glory, did not clothe himself like one of these. Very, very fascinating statement of our Lord's. This is looking at a very, very, very young plant, young flower. In fact, that's the shoot apical, that's Sam, in the centre there, and he's telling these things what to become. And this is now going to grow into a flower. It's a baby flower, and it'll grow, and then the petals and stuff, all those different parts. Yeah. And they're told what to do by Sam, and there's Sam in the background. He's telling this flower what to be, and then this one, and this one, and this one, and this one. And there's a lot of proteins interacting here. Yeah. Here's that SAM region again. It's where everything's organized. First of all, the cells grow, and you get a bunch of cells, and they're told what to become. They're going to make little flowers. It's really cool. The first thing that happens is a decision. Are you going to make a bunch of flowers or just one? And then once you decide you're going to make a single flower, well, which ring are you going to be? Are you going to be the petal ring, or the sepal ring? Are you going to be the stamen ring? And once that's decided, the next decision is, well, let's make a single petal. And all of those instructions have to be controlled. And these are some of the proteins which are involved in this process. And they don't work on their own. They all interact with each other. It's going to start looking like the London Underground soon. Communication, there's these different proteins and stuff. Amazing. Feedback loops, all sorts going on. Yeah. If this one gets mutated, boom, then we start to get plants that don't look right. Well, that plant doesn't look right. It looks as though it's too thick or something. We know, we can tell. Yeah? And in this particular case, it's this particular protein that got changed. This is the proper gene, and suddenly a bit was cut out. Just 5 base pairs, and now it doesn't work anymore. I know you get really weird flowers. You know. This is what SAM should look like, and this is what it becomes if those 5 base pairs don't work anymore. Too thick, and then you get inappropriate growth. So what's going on here? It's wise design, it's not mutation. The results prove an unfathomable number of beneficial mutation would be required to produce that. Remarkably sophisticated regulation of plant development by SAMs provides persuasive evidence of design. And mutations, they don't seem to have helped, they just spoilt it all. His 7th character was pretty straightforward. Sometimes he had a tall plant and sometimes he had a dwarf plant. Question, which one's better? Well, the tall plants, so some farmers, they like the dwarf plants because they don't fall over and they don't dry. So in that sense, it might be helpful. It's a tricky one, that. Yeah. What's happening is the uninteresting bit of the stem where there's no leaves hanging, it's called the internode. Yeah? And that's regulated in its length. And if it stays short, you get a really flat plant, and if they grow tall, then you get a really tall plant. So that's what's actually happening here. And this is regulated by a hormone. The hormone is called gibberellin. And if you want to make gibberellin, as the plant does, it's a multi-step process. First in one region, a 2-step conversion, and then a second, and then a third, and woo! Right at the very close to the very end, you actually add a hydroxyl group. Here's a little bit of that process, and you can see these different proteins. They have to be switched on at the right time, in the right sequence, otherwise none of it's going to work. It's irreducibly complex. And of course, if you make the hormone, yay! But you're going to need something to respond to the hormone otherwise. So you need a receptor, and then you'll need something that can react to the fact that the receptor's been... Loads and loads of stuff. This particular step, this is the protein which gets mutated in Mendel's dwarf plants. What happens? This is what the gene should look like, and then there's a change from a G to an A, substitution event. And that's where the gene's located on the chromosome 5, if I'm not mistaken. And that little change causes a 10-fold reduction in the activity of the gibberellin, the hormone. And so it's not working properly, so the plants are really tiny. They don't grow. Wise design, not mutation. Okay, so winding up. Because characters are alternative traits, the enormous potential for diversity is observed. Yeah. Usually recombination at meiosis, or possible combinations, they can give rise to new species. What's the difference? Well, we saw the difference. We saw those changes in the genes. 7 characters were studied, 9 actual sequences were investigated, and all 9 resulted from detrimental changes to the DNA sequence. Mutations degrade DNA. All 9 recessive phenotypes resulted in the dominant trait being compromised. It couldn't work properly anymore. The changes were 3 times there were substitutions, 3 times there were deletions, 5 base pairs, 400 base pairs, 100,000 base pairs. And 3 times they were insertions, 1,000 base pairs, 5,700 base pairs, and about 23,000 base pairs. Woo! Now, in 2 instances, one could argue it's not deleterious. But it's not getting any better. But it's just not deleterious. Yeah. Every protein component of a character, all of those proteins, they are extraordinary and precisely complex. If you look at these pictures of 3-dimensional pictures of proteins, they're crazy. They're just so incredibly well put together for exact specificity in action. But none of them would work unless they were part of a living cellular system. So we need all the buddies there. Otherwise, a protein on its own is just a waste of space. And of course, these characters, they work together to make the organism. how much irreducible complexity is in this kind of situation. Molecular consequences of genetic change, mutated genes, biosynthetic pathways were changed, gene regulatory pathways were changed. I was going to say some more about that. I will spare you that and come to these points. Any enzyme step in a biosynthetic pathway can be mutated. The thing isn't going to work. Any step in cellular signaling pathways can be mutated. It's not going to work. Any protein regulating the expression of genes gets mutated. It's not going to work. The complexity of the original traits, well, that's been known for some time, and it's always been a strong, persuasive evidence of design. New species can still arise due to the production of new derived traits, but they're derived traits, and typically we've lost something. We saw that very clearly. If we want ongoing genetic change, we can do it with things called transposons, we can do it with some stuff called epigenetics. Usually it's a mutation event. That's what was shown in Mendel's Ps. And that was good because it meant the species didn't become boring. There was still some change there. But it came at quite a cost because we lost very, very important stuff. Feng studies, this incredible study. What did it show? It elegantly compelling demonstrates speciation based on derived traits by DNA sequence change. But those changes are loss of genetic information. We're not getting more, we're losing it. It's as though we start with a lot and we can tweak it, but we're going to run out at some stage because there's no explanation for where something really cool and new and better might come from. In fact, they predict that the longer the time we have available, the more genomes will accumulate damage and they will just get destroyed. So conclusions and then the take-home message. In all 9 cases, the recessive phenotype resulted from loss of the dominant trait, not from something new. The evidence indicates recessive alleles arising from mutations are detrimental. Most of the changes to the DNA sequences have ruined the original good genetic information. Mutational changes to the DNA are in 0 of the 9 cases an actual improvement. This does not look, mutations do not look like a promising way to make organisms. The data confirm originally good trait information was deliberately lost to produce the plethora of fixed species, and current phenotypic trait production speciation result from mutating or tweaking existing information. Take-home messages: traits are derived from pre-existing characters when our Lord laid into the genomes at the start. And characters, they're never derived from mutations. There was never a single piece of evidence for that. They are irreducibly complex. They are the product of wise design. David recognized wise design early on. Wonderful your works. My soul knows it very well. Thank you for your attention, guys. Appreciate that.
Before we close, any questions you'd like to ask? Doug?
One thing I found very interesting is the design of chlorophyll. Very much is very similar to the design of hemoglobin. And except that one has magnesium in the middle, the other one has iron in the middle. One fixes oxygen, the other one fixes carbon dioxide. And I think the chemical pathways that create them are very similar. Am I correct?
Let's quickly move back to it, just to have a quick look again at that. So here we go. So this ring around the outside, very similar process to make that. Of course, hemoglobin doesn't have this tail. That tail is to make sure that it's sat in exactly the right position to capture the light. And of course, it has the heme in hemoglobin, that's an iron. an iron as opposed to a magnesium ion. Yeah. But yes, they are. They're very similar, and they're all irreducibly complex pathways.
And it's also an indication of a common design.
Yes. Yeah. Yeah. It's a great point.
Very dissimilar application. One is in animals, one is in plants. One, they sort of like the same design is used one to fix oxygen, one to fix carbon dioxide.
Yeah.
Amazing.
Yeah. I saw an article, and I'm trying to remember where I saw it, but it was recently, and it was about the Feng paper, and it said the heading was recessive genes may actually be genes being discarded or something like that. You know, and it was interesting to talk about this idea that maybe dominant recessive weren't original, but were something that had resulted from degradation.
Well, the recessive genes definitely appear to be derived. Yeah. The dominant, it's what all that irreducible complexity in the first place. Yeah, it's to achieve something, and then it gets tweaked, and then we have these alternatives. And the fact that it can be tweaked and can give us alternatives is nice because it gives us different species and stuff like that. Yeah, but it's not making anything new. Yeah, it's using what's already there. Yeah, so it's definitely something derived. Yeah.